we can get started. Good morning, and welcome to ClaretBio's quarterly webinar series, the Serious Talk. For those of you who may be a first-time attendee, I will just do a brief intro into ClaretBio. We are a small team based in Scotts Valley, California. We initially launched out of the UCSC Paleogenomics Lab in 2017, and we developed an NGS library prep method to help capture the maximum amount of genomic data from a variety of challenging samples. Originally applied for cell-free DNA, FFPE, and ancient DNA applications, our flagship product, Seriously, is a single reaction, single-stranded DNA library prep kit known for producing high molecular complexity libraries by capturing single-stranded, double-stranded, and nicked DNA templates without the need for end repair. In 2023, we expanded our method to RNA-Seq applications with the launch of our Really RNA Library Prep Kit, which our webinar will focus on today. For more information on our products, please check out our website or contact us at info@claretbio.com. As we progress through today's presentation, please submit your questions via the Q&A box at the bottom of your screen, and we'll dedicate the last 10 minutes to addressing those questions. Now, without further ado, I'm delighted to introduce our speaker, Andrew Mishin. Andrew is an integral part of Claret's research and development team and has extensive experience testing Claret's methods in the lab over the last three and a half years, helping to develop and improve our catalog products. He's a proud UC Santa Cruz alum, earning his undergraduate degree in biology. We're excited to have him share more info about the Really RNA-Seq library prep kit. Andrew, glad to have you here, and looking forward to your presentation on Really for whole blood and FFPE applications.
Thanks, Emily. Hi, everyone. My name is Andrew Mishin, and as Emily said, I'm the resident pipette head here at ClaretBio. I'm excited to be here with all of you on Hump Day to share our new Really hg-rNONE product. I'll go ahead and share my screen. Okay, and then does this look right, Emily? To you on your end?
Yep.
All right. All right. So, as I said, we're doing this webinar to mark our launch of hg-rNONE, and which is a hemoglobin depletion module that works in tandem with our existing Really rNONE kit, which is for making ribo-depleted RNA libraries. Now we're just offering a version of the kit that is compatible with globin-rich sample types. And before we jump into that new product, I want to give you all some background about our existing Really RNA-Seq technology. So on a couple slides ago, Emily showed a slide that had some challenges that people commonly face with DNA-Seq that we kind of sought to address with our DNA library prep. And all of those challenges still apply with RNA. So loss of, I guess, not single-stranded, but damaged molecules, loss of the RNA ends, and even more superfluous steps. The RNA protocols are generally long. But in addition to those things, the RNA is also especially prone to degradation. And if and when your RNA gets degraded, it can lead to bias and low complexity in your final library if you are able to make a successful library at all. So given this unique gauntlet of obstacles, Claret kind of wanted to leverage what we've already done with addressing degraded DNA for library prep and do the same thing for RNA. And I should note that our also kind of one of our focuses was not just to make a kit for just for degraded samples, but to make one kit that you can use for all your sample types without having to pick and choose which sample gets what kit. So we wanted to work well for everything. And here we have a just a broad overview of a comparison between a conventional double-stranded RNA library prep compared to ours, which is single-stranded. And I won't get too far into this, but what I really just want to show here is that because our product is based on a single-stranded technology, we have no need to perform second-strand synthesis, and we don't need to do any extra steps for making the library directional or stranded. That's all built in. So I like this diagram better, and I'll go through it with you all in a second. I just want to give a quick overview of the kind of perks as we see them of rNONE. So the Really rNONE kit generates high quality sequencing libraries from between 10 and 250 nanograms of high quality or degraded RNA input, and this can be human or mouse. Because, as I said, it's based on our single-stranded DNA library prep technology. No need for a second strand. We talked about the strandedness, less protocol steps. Also the RNA, the ribosomal RNA depletion happens after adapter ligation, and this allows for greater complexity and less bias as well. And at what we think is a pretty competitive price point. But so let me take you through the protocol. So the first thing that we do with Really rNONE is straight away we take our RNA and we put it into fragmentation for strand synthesis. And there is a modified protocol if your sample is already pretty degraded and you don't want extra fragmentation. There's a simple sort of tweak that you can do at this step. But I like this feature of the protocol, that this is the first step, because and I hope that if there are any other bench gremlins watching, they can also relate to this, that the less time I work with RNA, the better. So in this case, I have to be kind of careful in the first reaction. I'll RNA zap my bench, my pipettes, and then I'll set up my reaction carefully for first strand synthesis, and then I can kind of take a breath and not worry about breathing RNases into my tubes. So first strand synthesis generates our cDNA molecules, and that's done with a combination of random hexamers and poly-T anchor primers. And And then once you have your cDNA molecule, you denature it, heat denature to remove any secondary structures. And at that point, you have a perfect little single-stranded DNA molecule that is exactly what you would want to put into a seriously reaction. So that's what this step at the adapter ligation is. It's just our traditional seriously splinted adapter ligation. So if you've performed seriously, it's the protocol would seem very similar to you. And then after another bead clean, I failed to mention that there was a bead clean after cDNA synthesis, but so after the second bead clean post adapter ligation, that's when we have our depletion for ribosomal RNA. And that's performed via subtractive hybridization. So we just have a set of probes. Those anneal to the cDNA that has ribosomal regions, and we pull that out of solution with a bead clean, essentially. And then it's off to PCR. So all that in about 6 hours. And that's what we think is a pretty straightforward and common sense protocol without any extra steps. So now that we've talked about the protocol, let's talk about some of the various kinds of inputs that can go into really rNONE and the results thereof. So whenever you're doing an RNA library or an extraction in a subsequent library, there are many pitfalls, as I'm sure many of you know, but many potential pitfalls. But you can kind of generalize them into two. The first failure mode is just that you perform your RNA extraction, and immediately when you QC it, you see that by some indication, for example, your RIN value or RNA integrity number, you see that it's low quality immediately. And this is a big one because as far as I understand, in a clinical lab setting, up to 30% of samples can be excluded at this step just for low quality. But so the other 70% say, you have an extract that passes your internal QC metrics. You say, okay, this is good enough. This looks good enough to make into a RNA library. And then some of the time, more often than we'd like, for whatever reason, it still doesn't make a good library. And this can happen for a variety of reasons. Like it's definitely happened to me when I've, just haven't been awake enough when I started the library prep. But there are plenty of other reasons, specifically or especially with FFPE. There's a lot of variation in sample quality that you just can't see on like a tape station or a fragment analyzer or whatever you use. And this can be like stubborn crosslinks or chemical modifications from the formalin fixation process. But in any case, if there's something that you can't see that is making your RNA low quality, you may end up with a library that looks like this, or worse, even if it had a moderate RIN value, for example. But so we wanted to address each of these case scenarios, including the case scenario where you have a perfect extract that makes good libraries. So no sample left behind. So let's look at some of these samples now. So these are two examples of the two sort of most different case scenarios. On the left, it is a sample that was extracted from lung FFPE with a RIN value of 1.8, which is squarely in the range where a lot of people would throw this away or just not make a library out of it. But it is one that we've successfully made a library out of using really rNONE. And this is also the kind of sample that we would use that slightly modified fragmentation first strand synthesis protocol for that I alluded to before. And that change is very simple. It's just different incubation time, slightly different timing of adding the buffer, all very straightforward. But and then the one on the right is obviously a high quality total RNA, human RNA sample that we wish all our samples looked like. But so first, let's take a look at the worst case scenario. Probably the most interesting. Here we go. So what we're seeing here is some libraries that were made from liver FFPE RNA extracts with a RIN equal to 2. So just about the same as what we just looked at. And we did so with both the really rNONE kit as well as a double-stranded prep. We did it with 10, 50, and 100 nanograms, which is kind of the lower half of our input range. And what we see is that the really libraries look better overall, but even more so that the double-stranded prep in our hands didn't really make successful 10 nanogram or 50 nanogram libraries, or with that amount of input. And on the right, we're just showing that the concordance is good, even with these low inputs, and even when compared to the double-stranded prep or the one successful 100 nanogram double-stranded prep. But yeah, so we've established now that in our hands, the double-stranded prep doesn't really work for 10 nanogram or 50 nanogram inputs, but it does work for 100 nanogram inputs. So what would happen if we just compared some samples at that input level between really rNONE and double-stranded prep? So to answer that question, we did a few extractions with the kit that's written in the middle there, if anyone's curious. And we got three RNA extracts. And so the samples, 2 of them were cell line xenografts and 1 was a patient xenograft. All of them prostate FFPE. And as you can see on the right, lo and behold, the samples are degraded. And in a lot of places, I imagine this is kind of a bad day. But these are the kind of samples that I'm used to working with our tech. But so interesting to note here that extract 2 in the middle has like the lowest RIN ever, as well as of the three, but appears much larger in its average size than the other two, as visualized on a genomic tape station trace. And why is that? I don't really know. As I understand it, the RIN is calculated by a tape station based on calling of certain prevalent RNA peaks. So I don't know exactly why they're calculated the way that they are, but you can see that extract 1 and 3 are like 150 nucleotides long, whereas extract 2 is around 500, and that it kind of translates to the results that we're going to see better than the RIN does. And that kind of goes to show how complicated and how unique each FFPE sample can behave. But also, I should mention that we made these libraries with ERCC spike in, which is a high quality RNA that is synthetic, not from any organism. And it's just an internal control to make sure that, you know, everything in the library should have worked, to try to narrow down, is it the sample, is it a procedural error, etc. So all of these were prepped with that. So now we can really dive into comparing the metrics of both molecularly and sequencing metrics of Relay and the conventional double-stranded prep. So on the molecular side, on the left, so these are, again, tape station traces, D1, D1000. On the far left grouping is the Relay libraries. Immediately to the right is the double-stranded equivalents. UHR, which is the first trace in each section, that is a universal human RNA that's high quality that we use as another control, just to make sure that the library prep is working correctly, and that, you know, because we're working with low quality samples, how would a good sample look like when treated the same way? So from a molecular standpoint, and I'm looking down here at these numbers, specifically at the yield of the Relay libraries ends up being higher for all of these, including the high quality control, despite the fact that the double-stranded prep got 5 extra PCR cycles. And that is great for Relay and important for downstream complexity and the calling of the proportions of the various RNA species within the library. So on the right, then, are some of the mapping metrics from sequencing. And here, this can be a little confusing. Not everything is as it seems. So first, I'd like to point your attention to the far right section with the ERCC uniquely mapped percent. And before I do that, to orient you, we have the Relay hg-rNONE libraries in this claret red hues, and the double-stranded libraries in the blue-green teal hues. And they go, you know, the same way as the tape station traces go on the left, with the high quality control, and then sample 1, 2, and 3 for each grouping of bars. So back to the ERCC on the right here. So ERCC, to remind you, is that spike in that we did, the spike in control. And what you see is that for these 3 samples, which are sample 1, 2, and 3 For the double-stranded prep, the ERCC mapping is quite elevated. So why is that? The UHR, which is the high quality control for both really and the double-stranded prep, is down. You can't even really see it. It's at 0.1%, which is what we would expect with the amount that we spiked in. So that is the normal case scenario, and these being elevated is seemingly an indication of the fact that these poor quality samples, sample 1, 2, and 3, were the conversion rate into library was very poor. In this case, with this library prep. And so most of what you have in the final library is this control, because hardly any of the actual RNA we were interested in made it through to the library stage. And that is going to inflate the mapping rate as a whole here on the left. So for example, where am I? One, so this is UHR, this is sample 1, this is sample 2. So sample 2 here, this dark green bar is sample 2 for the double-stranded prep. That one is mapping at a rate of 90%, but if we go over here, like 50%, so half of this 90% is essentially junk. So and that kind of is corroborated by the duplicate rates here. You'll see that the claret red is kind of stays more subdued, lower duplicates, which is what we want, and while maintaining correct strand and robust depletion of ribosomal RNA. And here you could squint and see some differences between the double-stranded prep and really, but it mostly seems to be differences based on the sample as opposed to the prep. So for example, the sample 3 is particularly elevated, like 8 or 9% for both really and the double-stranded prep. And that just goes to show again that FFPE is, it throws you curveballs. You don't know looking at the extract how it's going to behave necessarily. So let me catch up here and make sure I didn't miss anything. Right. So bottom line here is that really did a better job seemingly at making libraries out of these low quality FFP extracts, even at an input rate at an input of 100 nanograms. But what if we use higher quality RNA instead? And what if we increase the input amount? Because we are trying to make one kit to work for all sample types. So when we try that and we use this same extract that we used here for as a high quality control, the UHR, we use it as the focus of an experiment instead at input ranging from 10 to 250 nanograms. Again, with both preps, both the really and the double-stranded. We see the same sort of thing molecularly where the yields are higher for really, even though it got 5 fewer cycles. So that kind of tracks and is the same as we've been seeing. And if we look at the sequencing results, again, we see the same thing where like mapping is good, but the duplicates are quite elevated in the double-stranded prep relative to really. And as a bonus, the really libraries had more uniform gene body coverage because, as we mentioned, we captured the 3' end, so we get this nice even curve here of coverage from the 5' to the 3' end of genes. So this is the same information presented another way with total unique reads instead of this percent duplicates. And I think this figures kind of speaks for itself, but I will point your attention to the fact that in this experiment, the complexity of the 10 nanogram input really library is pretty much the same as the 250 nanogram double-stranded library. And while maintaining, again, good concordance between across the input range from 10 nanograms to 250, which is the entire range that we support with really rNONE. So the bottom line here is that, like, overall, is that really just seems to generate higher quality libraries regardless of the quality of the input, whether it's the worst or the best quality input, and whether it's 10 nanograms or 250 nanograms. All right, the moment we've all been waiting for. So how does hemoglobin RNA depletion, how does hg-rNONE fit into this kit? So to reiterate, hg-rNONE is a new optional add-on to our regular really base kit that allows for hemoglobin RNA depletion in addition to ribosomal depletion. So we're just now making a version of the kit that's compatible with hemoglobin rich sample types. So what sample types are those? That's whole blood, erythroid cells, PBMC, bone marrow, biopsies. It can also be useful for spleen and liver, as those are highly vascular tissues. And this is, so you might think that, like, looking at the diagram on the left, that it looks really familiar, and you're completely right, because it's the same exact protocol. The only difference is at this ribo depletion step, you're also adding globin probes, and there's no extra time required except for that one pipetting step. All of the features and perks are all the same. We're just supporting all these new sample types now. So to show how this works, we did a simple experiment with 3 different sample types at 10 and 100 nanograms that mirror each other, but within each of these input amounts, we have undepleted, so no ribosomal, no hemoglobin depletion. Then you have ribosomal depletion, but no hemoglobin depletion. That's our regular, really rNONE kit that we've been talking about for so long. And then on the far right, in each trio, it's ribosomal and hemoglobin depletion. And what you see in both cases is a progressive improvement in the mapping of unique RNA reads. So when you introduce ribosomal depletion, you get rid of this red rRNA, and it looks like you have more hemoglobin RNA than you did before, but that's not really the case. It's just overrepresented because we removed all of this ribosomal RNA. But that's, that's still not good because it comprises 10% of your reads or more. So, and I failed to mention that this is whole blood RNA. So that's why you have that high of hemoglobin RNA. But so then when you do both ribosomal and hemoglobin depletion, most of the ribosomal and hemoglobin RNA is gone. You're left with a beautiful blue, green, teal. Somebody let me know what color that is in the comments. Bar with a bunch of unique RNA. And I want to point your attention to the fact as well that the ribosomal RNA stays low even when you're doing both hemoglobin and ribosomal depletion. So it's not that this kit, you get this kit and you get you have to get both the rNONE and hg-rNONE and decide which sample gets what. If you wanted to just have one kit for all your samples, like we were talking about, you can get the hg-rNONE kit and that'll work great for any samples, not just the hemoglobin rich samples. And so this is going into depth with the expression of hemoglobin genes with these same samples. And what you see is that without the hemoglobin probes, you see the kind of natural state of elevated hemoglobin genes. So on the left side, this grouping, this table has the more abundant hemoglobin genes, and note that the scale is different here. So this goes up to 1%, whereas this only goes up to 0.01% over here. But what you see is that in the green teal colors, you see robust depletion of both the more abundant and the less abundant hemoglobin genes, and while maintaining good concordance, like we've been saying. So that's the product. It's live on our website. Like most of our offerings, we have it at 24 reaction or 96 reactions. We have it with or without PCR primers. And you can still choose either the rNONE or hg-rNONE. And we're also offering Really Run for download from our website, which is our pipeline for RNA-Seq data analyses. If you need that, if you don't have that set up in-house already, that's on our website under software. And with that, I want to say thank you to everyone in attendance. And a special thank you to everybody, all my colleagues at Claret. We're a small team, and everybody did their part with launching this product. I'm really happy to have been a part of it, and I'm really looking forward to hearing from all of you in the mysterious internet audience. Thank you.
Awesome. Thanks, Andrew. If anyone has questions, please feel free to add them either in the chat or the Q&A box, and we can answer them.
Nice. That's the best kind of question.
Yeah, not a question, but good feedback. Yeah, if there are additional questions that come up, please do write to us, either at info@claretbio.com or contact technical support at claretbio.com. We will be sharing this webinar afterwards with attendees, and we'll post it to YouTube. We will be at some upcoming conferences starting next week or next month at ABRF. So if you happen to be there, please stop by. I'm happy to talk more about this in person. And yeah, that will conclude our webinar. Thank you.
Thank you.